All right, hey guys, it's chapter 15 time. We're getting close to midterms, so yay! All right, chapter 15 is about, lesson 1 is about the human chromosomes, and we're just going to go through the human genome a little bit more in this chapter. So the first thing, we've already talked about this a little bit in class, so karyotypes. Let's move this up here. Karyotypes show us the human genome. And this is a full set of genetic information that an organism carries in its DNA. The analysis of any genome starts with the chromosomes. So we know that this is showing us the homologous chromosomes. And we call this a karyotype. And what we are looking at with a karyotype is the complete diploid set of chromosomes. And they're grouped together in pairs. And they're arranged in order of decreasing size. And if you look at them here, you'll see that the 23rd pair that are the sex chromosomes, and those are the only two that will not be similar in size there. And this helps us to see if there are any chromosomal abnormalities or disorders in the person as well. Okay. So our sex chromosomes, there are 2 chromosomes that we call sex chromosomes. The female would have 2 X's, and males would have 1 X chromosome and 1 Y chromosome. We know that there are more than 1,400 genes found on the human X chromosome, and the human Y chromosome contains only about 100, I think that says 150 genes. It's cut off. But yeah, significantly less chromosomes. And this is showing you the X chromosome and the Y chromosome and what the different genes are and where they're located, because we do have a complete map of that now. So some of these are, lyssencephaly, that's like a brain disorder, and there's cardiomyopathy, which is a heart disorder, and color blindness would be on the X chromosome down here, different types of hemophilia, and we now know where to find all of these genetic disorders, and we'll be talking about biotechnologies soon and how gene therapies are made for them, different genetic disorders. Okay, so autosomal chromosomes, what that just means is it's all the other 22 pairs of chromosomes that are not sex chromosomes. So all of these 22 pairs are considered autosome. Okay, so and then we have our last pair, our 23rd pair, that are the chromosomes for sex, determining sex, male versus female. There are 46 total, and males, again, are XY, and females are XX. All right, so transmission of human traits. Many human traits follow a pattern of simple dominance. So, for example, the gene MC1R helps determine your skin and hair color. Some of the MC1R's recessive alleles produce red hair. An individual with red hair usually has 2 sets of these recessive alleles, and that is why we see the red hair as a physical trait. Okay. Codominant and multiple alleles. The alleles for many human genes display codominant inheritance. So phenotype of blood is the example that the book uses as showing us codominance or multiple alleles. So phenotype A blood, and then if you look at the genotype of those, they will have these antigens, capital A, on the outside of the blood cells. And they can give blood to anybody with A-type blood, A or B. And they can also receive blood types. They can only receive blood from other A people as well as blood type O, because blood type O doesn't have any antigens because it is the recessive trait. And there's also blood type B. So they can receive blood type B or AB from anybody as long as there's, they can give to blood type A, B, and B because they have it, and then have that B antigen. And then they can receive blood type B or O from anyone. Okay. AB is what we're talking about here when we say that codominant in multiple alleles. This is AB as in AB dominant, right? And then there's no, and you'll see here that there's a recessive allele there. So what you then get is a wide range of blood types that you can receive blood from. So if you are in need of blood, it is good to be blood type AB. You can receive blood from A, B, and O. So you're what they consider the universal recipient. If you are blood type O, you are the universal donor because everybody can receive your blood type. I know that I'm O positive and constantly being asked to give blood, because there is no antigen that would cause blood clots and coagulation. If you were given the wrong blood type, you would get blood clots and coagulation in your blood, which can ultimately lead to strokes. Okay. All right, sex-linked inheritance. The X and Y chromosomes do determine sex. The genes located on them show sex linkage. This image here is an example of color blindness. What you should be able to see is a number under each of these, but if you are colorblind, you might not be able to see this one here. That's for red-green color blindness. All right, so color blindness, like we saw, is on the X chromosome. So if you are a male, you have one X chromosome to give, right? So it is very likely that if you are a male with this color blindness trait, that it will be passed on to your son or daughter, but it's more apparent in males, typically, color blindness. Similarly, when we have X chromosomes, both sexes have an X chromosome. And we'll look at X chromosome inactivation now. So most genes in one of the X chromosomes are switched off, forming a region known as a bar body. So not all of the genes, because there are so many on the X chromosomes, not all of them are used. And this is the reason why female cats are typically calicos. Most calico cats are female because of that X chromosome and that bar body production. So the color of the spots on their fur is controlled by the gene that is only on the X chromosome. So because females have two X chromosomes, it's more likely for them. It's not impossible to have a male calico, but it is very uncommon because it is only found on the X chromosome. Human pedigrees. So we did a little bit of work on this last week, and we might do a little more practice, but a pedigree chart shows the relationships within a family. So a square represents a male, a circle represents a female. The vertical lines show the connectedness of the parents to their children. The horizontal lines show a marriage. And then the shaded circle or squares will indicate that that person expresses the trait. And a circle or a square that is not shaded does not express the trait. So in the book, we talk about this forelock trait. So this would be the presence of the forelock trait. That would be blue and filled in. So human pedigrees. Yes, I know it's a dog food, but no, we're not talking about dog food. We're talking about genetics. The information from pedigree analysis makes it possible to determine the nature of genes and the alleles associated with the inherited human trait. So we can see here, this is the father, and he passes it on to this child here, and then this child here, this marriage brought two parents together that have the trait as well, and then they ended up having one child. This parent here is showing you the wife has it and the husband doesn't, but then it's pretty common, so it seems like that is being passed on through the X chromosome. What do you think? All right. Okay. Human genetic disorders. is lesson 2. So chromosomal disorders, we call those non-disjunction typically. Non-disjunction is when homologous chromosomes don't separate as they should. So gametes with an abnormal number of chromosomes may result. So the homologous chromosomes will fail to separate properly during meiosis 1. So this one, should have been over here, right? Okay, and so what that ends up causing, typically, the most common one that we talk about is Down syndrome. The most common form of trisomy, and that's what Down syndrome is, a form of trisomy, involves 3 copies of chromosome 21. And you can see chromosome 21 right there. This would be a Down syndrome patient. And other chromosomal disorders that we'll talk about in the book is Turner syndrome. In Turner syndrome patients, they only the females only get one X chromosome, and because of that one X chromosome, they don't develop properly during puberty, and their reproductive organs don't develop, and they unfortunately can't have kids later in life. I actually have experienced this once in my lifetime. I have a twin mom friend that had boy-girl twins, and her daughter does have Turner syndrome, and it's been quite interesting to just see how they grow and develop, and she's a very sweet little girl. The other chromosomal disorder that we'll talk about is non-disjunction, causing Klinefelter syndrome. And that is where there is an extra X chromosome in the males, and the males will, it's a similar, very similar to Turner's, where they will not be able to reproduce later in life. And there is no cure or treatment for these type of chromosomal disorders. All right, so from molecule to phenotype. So changes in the DNA sequence of a gene can change proteins, and we've been talking about that, especially in the RNA chapter, altering amino acid sequences, and it affects the phenotype. And this picture showing you the example the book likes to use a lot is the sickle cell disease. So you can see that these cells here are the sickle cells, creating that sort of moon-like shape. Another genetic disorder we've talked about a lot is cystic fibrosis. Most cases result from the deletion of 3 bases in the DNA of a single gene, and the body doesn't produce the normal CFTR gene, which helps to break down the mucus in the lungs and disassemble it and properly. So if you don't have that normal CFTR gene, you have to do a lot of breathing treatments. There's a lot of things need to happen in order to remove the mucus from your lungs so that you don't get pneumonia and in essence like choke on your own mucus. Genetic advantages. So it's very rare, but there are some genetic advantages that having a mutation in an allele will help you. So carrying one copy of the sickle cell trait provides protection from malaria. So we'll read a little bit more about that in the book. And carrying one copy of the CF allele provides protection from typhoid bacterium was another study that we found. A benefit for having the CF trait, being a carrier for it. Okay, on to lesson 3. So studying of the human genome, and we watched the Cracking the Code of Life and kind of got to see the whole process happening way back then. So in order to look at the human genome, we need to manipulate the DNA. So we use tools that cut, separate, and then replicate the DNA base by base. And scientists now read the base sequences in DNA from any cell. And this is showing you the process of how a scientist will use E. coli, which is an enzyme, to cut the DNA strands at specific points in the DNA. So between the A and the G is where it's cutting, and it will make segments of DNA. And then they put it into this fancy machine here called gel electrophoresis. These are like jelly-like substances, and these are the wells, and you use a pipette to put the material in. And in here, this is where this picture happened, the cutting. And then they put them into these different wells, and it helps to separate the segments of the DNA according to the bases. Getting interrupted. Okay, so right here you'll see DNA fragments that are synthesized using an unknown strand as a template, and then we have these dye molecules here on the ends of each of the bases and where they were fragmented or cut. And what ends up happening is that the smaller pieces, like right here, see how this piece is smaller? It will move farther through the DNA. All right, and then and that's because it's going towards the positive end, because DNA is slightly negative, so it moves towards the positive end of this gel plate. And then it helps scientists to determine what bases and the segments of the DNA actually are, and you can match up the DNA to other segments. And then we can reassemble them, and we use computers typically to sort and order the fragments back out, and then we find the overlapping sequences and align them to determine the complete DNA sequence, to find how they line up and how it completes the entire genome. So that's how they did it. Cutting DNA, and scientists do that every day too. Cutting DNA, so DNA molecules must be cut into smaller pieces for analysis. Restriction enzymes are what we call them. That's what cuts the DNA molecules into fragments. Just like I explained on the last slide, it cuts them between the A and the G here, is what we're seeing, and we call these the sticky ends. Those longer overlapping segments are called sticky ends. And there they are. Okay, so separating the DNA, we use, like I already explained, gel electrophoresis. It's used to separate and analyze the differently sized fragments of DNA. So here's your electrical equipment. There's a negative end here and a positive probe here, so charge is running through it, and it sits in a liquid. And this is made of partially agarose gel, and the DNA fragments will move through it. And we're going to be looking more into these biotechnology techniques this week. All right, so reading DNA. So a single-stranded DNA fragment will be placed in the test tube with the DNA polymerase and the nucleotide bases A, T, G, and C. And that helps us to determine what these different bases are, because we know that this one will move further, and they'll know the size of the fragments, and they'll be able to compare them to an unknown sample, for example. All right, and so that helps us to determine the different bases. Okay, so assembling the sequence, we use something called shotgun sequencing. That's where the chromosomes are cut into fragments, and the fragments are sequenced, and the information is then fed into a computer. So this is just walking you through it at a slower pace than I basically explained all this on the first slide we saw. Okay, so the human genome, what's inside? In 2003, the first complete human genome was sequenced. We get to see that happen in the video. Labs around the world now study which regions of DNA are being whatever's necessary. And we are transcribing them into RNA. We are binding to proteins. They are marked with epigenetic tags, and they vary from one individual to the next. All right, so how many genes? So human cells have approximately 20,000 genes. Scientists don't know the function of more than 25% of these human genes. There's a large portion. The large and small of it. So the human genome has 3 billion bases. Only about 2% codes for proteins. And this is just showing you the different genes in the different organisms. All right, so the personal genome and genome privacy. Comparing two humans, only about one base in 1,200 will not match. We are very genetically similar. Increasingly rapid sequencing technology is making it easier to identify these differences, and that's when we discussed 23andMe and how you can find out your genetic ethnicities already, like pretty quickly, and as well as ancestry and stuff like that. And people are saving all this information on databases now. You could also find out your medical genome. If you want to know genetic disorders, there's a variety of tests that companies like 23andMe will do now. But there are some legal and ethical questions about the human genome and how we will use it in the future. And we will discuss that in class, and we did already a little bit. Okay, so gene imprinting. Due to epigenetic marks, some genes are only expressed if they came from a male parent, while some genes are only expressed if they came from a female parent. So this is showing you Angelman syndrome. So Angelman syndrome, the chromosome 15 from the father is silenced, and the chromosome 15 from mother has a gene deletion. And that means that Angelman syndrome will be expressed, and this is just, there's muscle issues and development issues and things like that. So the child doesn't develop typically, is how they would refer to it. And on the other end, though, a normal phenotype, so chromosome 15 from the father has the gene deletion but is silenced, and the chromosome 15 from the mother has an active gene. So there's other ones that are gene imprinting or through epigenetic marks like this one. Another one is called Prader-Willi syndrome, and I've had one student while teaching that had Prader-Willi syndrome, so it's pretty rare, but very interesting that it is because of silencing of a gene. So, all right, and that's just showing you the difference between the normal phenotype and the Angelman syndrome. All right, so that concludes our chapter 15. I will be putting out chapter 16 shortly, and then we will be having a midterm. So let me know if you have any questions in the comments below. Bye, guys.